Okay, as usual, do you see the screen?
Yes.
Yes.
Okay, so as usual, we start with this Kahoot quiz. Okay, let's start. Okay, today we have 6 questions. Okay, perfect answer. Very good. So bacteria has a cell wall, and outside of a cell wall, there are cell membrane. Hey, you should say something. That's not true. Cell wall is outside of cell membrane is outside of a cell wall.
No.
No. So cell wall is outside of a cell membrane. Cell membrane is inside. And the bacteria has the genome. That means it has the genetic information, the whole set. And also it has a plasmid. But bacteria does not have a nucleus. So no nucleus means it does not mean that it does not have a DNA. DNA is there, but just that there is no membrane, the capturing, enclosing the DNA inside. Okay, only the nuclear membrane is missing in bacteria. Okay, very good. Move on. Dongwoo was the fastest. Okay, second problem. Mammalian cell. Mammalian means it's a 포유류. That means it's an animal cell. Okay, majority of you had the right answer. Maybe the question was a little ambiguous. What is unique in mammalian cell? That means something only in mammalian cell. Okay, maybe you misunderstood the question opposite way. So we don't have a cell wall. We have only membrane. And we have mitochondria. Mitochondria is originally from, we believe, it's from some different kind of bacteria. So mitochondria has a special bacteria living together with us, we can think. So other bacteria, they don't have a mitochondria. They do have a mitochondrial function on their the plasma membrane of bacteria itself. Okay, plasmid is only in bacteria. We don't have a plasmid. Cell membrane, we do. Bacteria do. How about the virus? Virus has a the cell membrane. Yes. Nope.
Yes.
Yes. All of them. Cell membrane. Okay, first of all, some bacteria, they don't have this cell membrane. And also, even though they do have this lipid structure, they don't call it cell membrane. They call it envelope or lipid membrane, but they don't call cell membrane because it's not cell. And many of the bacteria, actually as a virus, they do not have this the membrane structure. They are just have protein structure capturing those DNA inside. Those protein, do you know how to call that kind of protein? Virus, we call protein coat. Coat is wet too outside. So protein coat we call, but envelope means it's a membrane. But that membrane is not the protein. So some virus has the membrane, but that is called as envelope. Okay. many viruses, they do not have this membrane structure. They have only the protein coat structures. Okay, move on. Okay, Sejin is coming in. Okay, great. So this is just a checking up. This is a primary school, the questions. Whether, okay, welcome, Sejin. Do you hear me?
Yeah, I can.
Okay, so join us to this Kahoot quickly. So our main, the three major building blocks of our body, we have learned it's a protein, carbohydrate, and lipid. And vitamin is kind of a very small amount of material, which is necessary, but it's kind of a catalyst or cofactor. We don't call it as a building block. Building block means it's some composition of our body. So I just wanted to remind you, these three are the main building blocks, and the real problem is in the next. Okay, this is also checking the basis. Cell membrane is a lipid structure. And then you may expect what is the next question. I don't have a Sejin here. Sejin, you know the pin number is here, right?
Yeah, I joined it.
Okay. I'm not sure whether this is the last problem or not. Okay. Oh, this is interesting. I thought this is a quite easy question. Actually, this was the only the real question for today's the Kahoot. What is the structure of cell wall? Bacteria has the cell membrane, but outside of the cell membrane, there are some another structure of a cell wall, right? So this is cell wall. What is the name of that structure? Peptidoglycan. Peptido means peptido is a peptide, or if it's a little big, then you can call this is a protein. Small protein is a peptide. Okay. Glycan. What is a glycan?
Carbon.
Glycan means carbohydrate. So only bacteria has cell wall. We do not have a cell wall. So if we develop drugs which can selectively attack only bacteria, then this cell wall can be good target. Because we don't have a cell wall, so some compound attack this cell wall, then our body may not have any side effect. That makes sense, right?
Yes.
So bacteria is coming and invading our body, and they compete with our own cells. They do not come into the cell. Usually they live outside of the cell. And then they live outside of the cell. That's why they are competing with us. And one good chance is to attack some structure we do not have. Then that's a safe approach to attack, not killing our own cell, but we just kill only the bacteria. Okay, so that's why I guess this is the last. One more question. Okay. Okay, very good. So here I tried to give you some part of it, some part of the targets of antibiotic. Antibiotic, last time we said this is antibacteria, is antibiotic, right? So penicillin and vancomycin, they attack this cell wall. And antibody, we didn't learn it yet, but antibody is huge, it's very big. It's bigger than the thickness of a cell membrane, but antibody is too big. That's why it's difficult for the antibody to enter the cell through the membrane. So that's why we can assume that bacteria outside the surface is the cell wall structure, and antibody's first chance to attach is the cell membrane, the cell wall, not cell membrane or inside. Okay. Now, we briefly discussed the last time the mechanism of antibiotics, and one of them was a cell wall attack for bacteriocidal. What is the other mechanism? Do you remember?
Bacteriostatic.
Say again.
Bacteriostatic.
Bacteriostatic is the result of the action of the drug. What was the mechanism? What is the target of those antibiotics?
Enzyme.
Enzyme. Did you say enzyme? Okay, enzyme. Maybe some specific the bacterial enzyme can be target. That's one possibility. But we said some other terms.
Inhibiting metabolism, DNA
Metabolism inhibiting was one of them. Maybe that's that can be the enzyme. What is the other?
DNA replication.
DNA replication.
DNA replication, DNA synthesis. That can be another target. Now this quinolone is targeting DNA synthesis. There was one more. What is it? Protein synthesis, right? So when I give out the assignment this week, many of you decided to go for penicillin. 2 of you selected the vancomycin. 2 of you selected the quinolone. So there was no examples for the other 2 mechanisms. So we are not going to talk about much those other mechanisms, but today we are going to mainly focus on cell wall attack by penicillin and vancomycin and quinolone for DNA synthesis. Okay. Now cell wall synthesis, as I said, this structure we do not have in our body. That's why if that drug is selectively attack cell wall, we are okay because we don't have them. Now, DNA synthesis is a little different problem. DNA synthesis, we need the DNA synthesis too, then there should be some other selectivity mechanism. If not, that drug can attack our own body too, right? So that kind of examples we are going to hear today. So let's see. Okay, so today's class is the focusing on the antibiotics. We just finished this quiz. Now, I understand this course is for the sophomore, and I do not require, many of you has very strong organic chemistry the background, but today we are going to see some of those organic the molecule structure. So that's very briefly we are going to discuss about this. Now, organic molecules. What is organic molecule? How do you define organic molecule? Let's give a chance to the sophomores first. Someone, what is organic molecule?
Molecules that organize life.
Organizing life. Okay, here the organic means this is generated from life or generated for life. That's organic. Okay, organic does not mean that we did not use a pesticide. Organic means originally it's from life. That's organic. So many people believe that previously this organic molecule can be generated only by life, for life, but sometime later we realized then these organic molecules are just chemicals. We can synthesize them too. So that's why it becomes part of our synthetic the chemistry the field too. Then we need a different definition for this organic molecule. Then structure-wise, how do you define? How can you define organic molecule? Doyon?
Organic molecule is that consisted with C carbon
Consisted
and
of
hydro. consisted of CHO and etc.
CHON, Doyoung gave 4 of them. Which one is essential?
C and H.
C and H is essential. So you may put up to the C first, compound with a carbon, molecule with a carbon. So that makes sense. But carbon itself, let's just say just carbon itself can be either diamond or coal. Those are not organic molecules. Carbon plus oxygen, CO or CO2, we don't call it organic molecules. So organic molecules is based on carbon, but they should have a hydrogen together. Then that's a basic the requirement for organic molecule. And oxygen or nitrogen is optional. Some of them they can have oxygen, some of them they can have a nitrogen or phosphorus or other structure. But basically, organic molecule we can call the hydrogen and carbon containing hydrocarbon is organic molecules. Okay. Now some examples of organic molecule, let's just say, then this molecule. What is this molecule? Youngman?
Ethanol.
This is ethanol, right? Ethanol can be, if you draw those structure, maybe you can draw the structure like this way. Is that correct? Chemical, the bond structure, is the correct structure? Is this ethanol? Yes? No? Something wrong? Strange?
Oh,
correct?
wrong.
Wrong? Which part is
no.
wrong?
I don't know. Correct.
This is correct. So we see here the 1 carbon, here the carbon, and the carbon has a 4 hand. So this is connected here and here, 1 each of 3 hydrogen and 2 hydrogen here and OH. Maybe you can draw bond here or not, that's optional. So this is another way to draw ethanol. But problem is, this is quite a small structure, so that's okay. But if it becomes bigger and bigger, then if you should draw all of this hydrogen, then it's a little too much work, right? So that's why for the bigger molecule, we have different notations. We just draw only carbon structure. And then if you don't draw anything, then we assume that that is occupied by hydrogen. But that hydrogen, we don't omit if it is connected to heteroatom, like oxygen or nitrogen. So this is the simplest version of drawing organic molecules. Okay, so if it's becoming bigger and bigger, just only 2 carbon molecule. This is a relatively simple organic molecule. If you need to draw all the hydrogen like this, it takes too long time. So to make it easy, we take this approach. So then we need to be familiar with this kind of notations. Okay, by looking at this, you should be able to say, oh, this is ethanol. Then what is this molecule? Is it difficult?
Methanol.
This is a methanol. This is a 1 carbon here, and then connected to OH. Looks a little weird, but small one is more strange. Bigger one is easier. If you replace this with this hydrogen, then what is this molecule?
Water.
This is water, right? So water, you can optional, you can say H2O, or you can draw like this if you want to point out these structures. So this kind of structure, if it's small, looks a little weird, but if it becomes bigger, like this, how many carbon here?
7.
7.
7. If it's 7, then what do you call this molecule?
Septanol.
Sep. Sep.
Hep.
Heptanol.
Hep.
Hep.
Hep is 6, isn't it?
Sep.
Sorry. Hex is 6. Hep is 7. Sorry, I'm wrong. This is a heptanol. So it's a 7 other carbon structure. Now, to draw all of those hydrogen is a little complicated. So usually we draw this way. Can you count the carbon number?
7.
7, is that correct? 1, 2, 3, 4, 5, 6, 7. You don't count this, right? This is connected to the oxygen. So by looking at this structure, you should be able to think out, oh, this is heptanol. Okay, so this notation we are going to use from now on. Okay, so it may take a little time, but you will be familiar with this notation very quickly. Now here, if I change this OH into this carboxylate group, then maybe you can draw this way. Is this the right structure? Converting this into this? If it's wrong, then what is wrong?
Number of carbon.
Number of carbon here is what?
7.
7, including this, right? This carbon is here, and then I draw this part like this. This is your OH, so 1 carbon is missing. Someone, is that clear? So then I needed to put 1 more carbon here. Then this is the right structure of this. Okay. You need a little practice to make yourself familiar for this. Anyway, this is a notation we are going to use from now on. Now here, one thing I want to talk with you. Now here, this is a carbon and this is a carbon, right? And what is the length or what is the distance of between this carbon to this carbon? Carbon to carbon length. In this class, I do not require exact number. Okay, in this class, I require you approximate scale. 50%, 200% error is fine if it's the same order. Okay, I'm asking you whether this is 1 centimeter or 1 kilometer. I'm not asking you this is 1.035 per centimeter. Distance between carbon to carbon, what scale?
120 picometer.
Picometer? I heard 5 answers at the same time, so I couldn't recognize them all. You said something picometer. What picometer? You guys say one by one. Doyun, what did you say?
10.
10 picometer?
120.
120 picometer? Everybody agree? 120 picometer? You know, for chemistry, so those picometer or nanometer, those unit is not very convenient. But you know, most convenient the method may be in the 1, 2, 3, 4 range, right? From 1 to 10. This range is more easier than 100 or 0.1 kind of things, right? So then that's why we invented another non-standard unit. What is it?
Armstrong.
Angstrom. Yeah, Angstrom. So Angstrom, this the unit here, Doyun's answer is close. 120, actually this is 150. C and H distance is about 120. So this distance is 150 picometer. That means 0.15 nanometer. So that's a little not convenient. That's why we call this 1.5 angstrom. So 1 angstrom, 10 angstrom is 1 nanometer. Okay, so depending on those atoms in between, it can be a little longer and a little shorter. But the shorter one, like hydrogen to carbon, this is about 1.2. Hydrogen to hydrogen, that may be also a little bigger than 1 angstrom. And if it's a big atom, like phosphorus, still that distance is not longer than 2 angstrom. So that means it's similar to the human size. Most of us is between 1 meter to 2 meter, right? If it's adult. Similar, in this angstrom, most of those atom distance is about 1.5 and a little bigger or smaller, more or less. Okay. Then, if you think about those distance from here to here, maybe let's just say up to oxygen, then how long is this? How many bonds in between? 8 bonds, right? 1, 2, 3, 4, 5, 6, 7, 8. Now, this is not stretched out, right? This is a zigzag shape. Then approximately this one, you can say, if this is 1.5, approximately this distance is approximately 1 angstrom, we can assume. It's a reasonable assumption, right? Then if it's 8 bond, then we can say approximately this is 8 angstrom. 8 angstrom is 0.8 nanometer. Okay, this kind of approximate the thinking can give you the general scale of those molecular the size. Okay, so using this, let's see. So let's look up at a little more the different structures. This molecule, do you know what is this molecule?
Glucose.
This molecule, I don't know whether this is glucose or not. But anyway, this is a carbohydrate. So hydrocarbon is a carbon and hydrogen is a hydrocarbon, hydrated carbon, that's a hydrocarbon. Now there's a lot of hydroxyl group is attached. Then this kind of a molecule we call carbohydrate. Hydrate means water. That means water added hydrocarbon is a carbohydrate. Okay, any kind of organic molecule with a many hydroxyl group that can be defined as a carbohydrate. So that means 물질이 많이 된 유기분자 is a carbohydrate. In our the language, 탄수화물, 탄 is a carbon, 수화물 is hydrated. It's exactly the same translation, carbohydrate. Okay, any molecule with many hydroxyl group, that is a carbohydrate. Now, if the molecule has many hydroxyl group, they may behave like a water miscible, right? Hydrophilic molecules. And this structure can be drawn in a different format, like this and this and this. All of the molecule is the same molecule. Yes, this is a little difficult to recognize, but if you draw this way, yes, this is a glucose. So depending on how we want to draw this structure, all of them is the same molecule, but we have a little different way to draw these molecules depending on for the convenience. Okay. At your level for now, you don't need to be able to draw this, but at least by looking at those structure, oh, this is a carbohydrate structure. If you can recognize it, that is good. So carbohydrate was one of our main building block of our body. What is another building block? We talked the 3 building blocks, right? Carbohydrate and protein. Protein's building block is this kind of structure, amino acid. This is amino acid structure. Why do we call it amino acid? It has an amine group here, and then the other side we have acid here. So by looking at those structure, amino acid. This is the name of the origin of the name of this structure. And this amino acid can be combined together, one amino acid and the other, and then this carboxylate and amine group can be conjugated together and condensed. When you said condensed, by removing one water molecule and they are connected together, and this is a condensation reaction. And this is the way we can make, by removing one water, we can connect amino acid one by one like this. And if this is peptide, peptide means from 2 up to maybe 9 amino acid connected small protein, we call it peptide. And if it's bigger than 10 or 20, now we start to call it more protein than peptide. So basically, amino acid is a single building block of peptide or protein. Peptide is a small protein, protein is a big one. Okay, this is another building block of our body. And the name is also clear. Now from here, amino acids. Okay, how many amino acids do we have in our body? Basic amino acid to make a protein. Never heard?
26.
About 20, 20,
26.
26. Wow, is it? When I learned this biochemistry, the basic amino acid is 20, and there is some exceptional ones, small exceptions. Not every life form. Some bacteria, they have a little different the forms. Like here, rather than the sulfur is selenium or different structure. So 21, 22, 23. I recall I heard up to maybe 23 kind of amino acids, but common amino acids is except those kind of exceptional ones. Common amino acids are 20. So this 20 is the main building blocks to make our proteins. Okay. What is the other third the building block of our body?
Lipid.
Lipid. Lipid is a long hydrocarbon structure like this, and many of them is not only just hydrocarbon. If it's just a simple hydrocarbon without this structure, without this acid, then that is more like a gasoline. Okay, so those gasolines we cannot eat. That has a lot of energy inside, but we cannot eat and digest. Because of this acid group added at the end, that's why we can eat it and we can handle it. At least we need to have some hydrophilic motif of long hydrophobic carbons. How many carbon here in this molecule?
18.
18.
This is 18 the carbon. And then what's the length of this from here to this oxygen?
18 ohms.
18.
Is it 18 or 19?
Oh,
Oh,
19.
are you sure?
17.
18.
17? Many different answers.
18.
It's 18 because the atom number is from here to here is a 19. So we count those in between these, right? So it's 18. If it's 18, then what is the length?
18 angstrom.
18 angstrom, 1.8 nanometer, right? So if you make this molecule the many, then they will make this kind of a layer. If you have another layer in another side, then these layers, this side is hydrophobic to each other. So they meet here, and then hydrophilic the surface is going to the outside. They make a big layer. This is the lipid bilayer structure. So of course the head group is a little different in real the cell membrane, but similarly one side is a hydrophilic group, and then this side is a hydrophobic in the middle. Then lengthwise, this side you said 1.8 nanometer. Now we have a double layer. Then what is the total length from here to here? It's the thickness of membrane. What is the thickness?
.6 nanometer.
This is the rationale that the membrane thickness may be around the 4 nanometer. Okay, so majority of those lipid molecules, the composition, it has maybe 18 or 20, that kind of the range. That's the highest, the most popular the lipid length, and it's a double layer. That's why one side is about the 2 nanometer, and we have a double the layer of it. That's why the whole thickness of a membrane is approximately 4 nanometer. Okay, so that part we will come back later again, but this is good the gauge. You may need to keep it in mind. Then you can compare this thickness with the different interacting molecules. Okay. Okay, so for today, I invited the 4 people for the mechanism of antibiotics. Some of you made a better the presentation material, but depending on those topics, I want to focus on which topic is complement to each other. So I invited Doyun for PNC. This is mainly about the penicillin, the basic penicillin the part. What is the PNC, Doyun?
Penicillin.
PNC means penicillin? Or it has some specific penicillin? Anyway, Doyun is going to talk about the penicillin. And some of those penicillin story was missing, so I invited someone a little more modified penicillin derivatives with a little different the mechanism of resistance. So these two is targeting cell wall synthesis. There was another molecule, vancomycin. It's a totally different structure, but target is the same, the cell wall, but the mechanism is different from penicillin. So we are going to compare those mechanisms with this. And then 2 of you also they gave the quinolone. This is a targeting DNA synthesis. And we are going to study about this by hearing one by one for this. Okay. So Doyun, now it's your call. So go ahead. You may share your file. and then we talk, we hear your talk. Okay, I can see your screen. You may start.
Okay. Hello, my name is Kim Do-yoon, and today's topic is, my topic is about penicillin. And the contents are why I chose this topic and discovery of penicillin and mechanism of PNC and resistance for PNC. But actually, I made PPT on discovery of PNC. It's a broad PNC, I think, and mechanism and resistance for PNC is for beta-lactam antibiotics, I guess. PNC is one of the beta-lactam antibiotics. The story of penicillin discovery is recorded in a middle school science textbook, and it demonstrates that PNC is very famous antibiotics than other drugs relatively. However, its mechanism and resistance for it is not exactly known. For these reasons, I choose this topic to satisfy my curiosity. Fleming got summer vacation in 1928, leaving his Staphylococcus in Korean 포도상구균, and that played outside. Downstairs of the Fleming's lab, another scientist was researching on the specific mold, which can make the material that kill bacteria. And Fleming realized that PNC was discovered right after he arrived home. What happened during his vacation? The mold react with the colony, and the colony size reduced where the mold exists. If the Fleming is very lucky because if the mold weren't that specific mold, PNC's discovery would be delayed. Here is mechanisms of PNC. PNC is inhibitor of trans-peptidase, and additionally PNC is a competitive inhibitor. Trans-peptidase is an enzyme that help binding peptidoglycan cells together. And this picture is how PNC inhibit the trans-peptidase. First, without PNC, trans-peptidase can help peptidoglycan cells binding, but this orange color P initial stuff is penicillin. Penicillin is binding with trans-peptidase instead peptidoglycan cells. So peptidoglycan can't bind with that trans-peptidase and enzyme. Then weaker and weaker cell wall of bacteria. Eventually, tremendous osmotic pressure changes, and peptidoglycan will die. So PNC is bacteriocidal. Now resistance for penicillin. PNC molecule has beta-lactam ring, which consisted of 3 carbon and 1 nitrogen atom. And PNC is one of the beta-lactam antibiotics that have beta-lactam. Beta-lactam can bind to active site of trans-peptidase, so it can work. But bacteria have made the material beta-lactamase, which destroys beta-lactam structure. Its mechanism is on the picture. So beta-lactamase can, this 3 carbons and 1 nitrogen square ring will be changed by beta-lactamase. Then finally, they can't bind, so penicillin is not working. Thank you.
Okay, thank you, Doyun. Question 2, Doyun. A question about this mechanism of antibiotic.
Excuse me.
Any question for Doyun? Everybody is clear for the penicillin activity? Okay, then let's hear the Sungwon's the follow-up, and let's talk about it combined. Sungwon, would you start?
Our presentation about amoxicillin, one of the penicillin antibiotics. This is the table of contents. Firstly, what is amoxicillin and discovery of amoxicillin and mechanism of amoxicillin and amoxicillin resistance and side effects, and lastly, noted items of amoxicillin. Amoxicillin is the oral penicillin antibiotic, and it is very generic medicine that affects on both gram-positive and gram-negative bacteria. It can treat with middle ear infection, strep throat, pneumonia, skin infections, salmonella infections, etc. It listed on World Health Organization's list of essential medicines, the safest and most effective medicines needed in a health system. Then, how amoxicillin discovered? Firstly, 1924, Alexander Fleming discovered penicillin, the first antibiotic drug. This saved many lives from bacterial infection, but this was the narrow spectrum antibiotics, only for gram-positive bacteria. So scientists discover ampicillin that treat a broader range of both gram-positive and gram-negative infections. But this ampicillin was not that longer, so this amoxicillin is discovered. By having an additional hydroxyl group on benzene ring, amoxicillin is more lipid soluble in comparison ampicillin. By this reason, amoxicillin can action for long duration. Before explaining mechanism of amoxicillin, I will explain you PBP and beta-lactamases. PBP is penicillin binding proteins that transmembrane surface enzymes which catalyze the cross-linking between the peptidoglycans in the bacterial cell wall. Simply, PBC is an enzyme that catalyzes synthesis of bacterial cell wall. And beta-lactamase is the hydrolase that catalyzes this reaction, which destruct beta-lactam, so eliminate antibacterial function of drug. Amoxicillin inhibit bacterial cell wall synthesis by geometrically mimicking this substrate that is generally bound to PBPs. Bacteria cannot synthesize their cell wall, so bacteria die due to lysis. And in comparison with penicillin, amoxicillin have special mechanism that combine with clavulanic acid, one of antibiotic. By this mechanism, beta-lactamase is deactivated, so this drug acts their antibacterial function longer. There is 4 general mechanisms of amoxicillin resistance. Firstly, inactivation of antibiotic by beta-lactamase. As I explained already, beta-lactamase catalyze the destruction of beta-lactam ring by hydrolysis. So this ring become penicilloic acid and antibacterial function disappear. And secondly, excessive efflux of amoxicillin from bacteria. There is the region between the plasma membrane and the outer membrane or cell wall. A periplasmic space. And in bacteria's membrane, there is efflux pump, and this pump efflux the amoxicillin from this periplasmic to outer membrane, so this drug can act in bacteria. Thirdly, impaired penetration of drug of target PBPs. This occurs in gram-negative bacteria due to impermeable outer cellular membrane. So the drug can act too. And finally, modification of target PBPs. As I explained, PBP is penicillin binding protein and catalyze synthesis of cell wall of bacteria, and the penicillin or other amoxicillin or other penicillin antibiotics target this protein and inactivate cell synthesis. But if target PBP have modification, this reaction will not work. There are several side effects of amoxicillin.
Okay, Sangman, I think this is fine. Then let's stop here and let's review. The major part was already covered. Now, do you have any question to Sangman or the Doyon about the penicillin or modified penicillin the structures?
I have a question.
Yeah, go ahead.
I think the amoxicillin is quite special because most of antibiotics is used for only for either gram-positive or gram-negative because of their different mechanism or different structure of cellular. But in case of amoxicillin, you told that it is working for both gram-positive and negative at the same time. How is it possible? Is there any special mechanism for amoxicillin which can make it possible?
Sorry, I forgot.
Okay, that's fine. For now, Sangman, would you take out your slide? I want to use my slide.
Yes.
Okay. Gram-positive and negative, the mechanism difference, it may take a little more time. So that part we'll talk later. So let's come back to this original picture here of there are many things here. Okay, original. So this was a Doyun's the slide. You can see them, right? Everybody, you can see the Doyun's slide.
Yes.
Okay.
Yes.
So here, this is, let's just say this is representing gram-positive bacteria, which has the cell membrane inside and the outside is the cell the wall. Cell wall is actually more penetratable, but it has this kind of a cross structure. That cross structure is this is a carbohydrate, and then those carbohydrates is connected by peptide. To make this peptide cross-linked, there is some enzyme they need. So this enzyme is a penicillin binding protein. And this enzyme's function is a trans-peptidase, means by changing this structure, they connect this structure. So this strand and this strand right now, it's not connected. Using these enzymes, they make a cross-linking of this. If it is cross-linked, then this cell wall is fully structured, and then they can act as a protection for bacteria. Penicillin binds to this, and then it inhibits the enzyme function over here, and then they inhibit this cross-linking. That's why cell wall cannot be properly synthesized. So that means penicillin's main function should work against mainly gram-positive bacteria. Now, gram-negative are the bacteria. Those differences we will cover later. So for now, let's talk about the gram-positive bacteria. Cell wall is a target of this penicillin. That's a key part. And for binding to those the penicillin binding protein or the enzyme, this structure is essential. This structure here, 4-membered ring of the amide bond here, amide or the peptide bond with the 4-membered ring. This is a so-called beta-lactam. Now you can assume that then if it is a 3-membered ring, the the triangle structure and then lactam, then how do you like to call? That is a alpha lactam. Okay. If it's a 5-membered ring, how do you like to call? Alpha, beta, what's next?
Comma.
It's a gamma lactam. If it's a 6-membered ring, you may call delta lactam or something like that. Okay. It has some system, alpha, beta, gamma, delta. Anyway, this 4-membered ring has a lot of strengths. That means it has stored the potential energy inside. It's kind of almost exploding because of their strength. Okay, this structure is quite unique, and this is the essential part to bind to this protein, so-called the penicillin binding protein. This is active enzyme which make those peptidoglycan connected together. Okay. So this is the mechanism of penicillin. Amoxicillin has a little modified structure to avoid, to extend that activity to gram-negative. That is more access issues rather than the mechanism issue. And resistance. So during the first presentation, if this is 4-membered ring is important. If a bacteria develop some enzyme which can break out this bond, this reaction is a hydrolysis. Hydrolysis is by adding water and you destroy the bond. So by adding water, this one and this one, and if you reverse this reaction, this is similar to peptide formation from acid amine group conjugations, from the amino acid conjugation. Okay, that reverse reaction is hydrolysis. So one of those mechanisms for resistance for bacteria is to destroy the penicillin, destroy the beta-lactam structure. That's one mechanism. Okay, this is what Doyun others showed, and Seung showed another mechanisms. So key part here, she repeated this hydrolysis. This is one mechanism for bacteria. I don't like penicillin. Penicillin or beta-lactam, they block our cell wall synthesis. If a cell wall is not properly made, cell wall just provide, cell wall is kind of a permeable structure. Water can penetrate the cell wall. Cell membrane is kind of a blocking the water access. Cell wall, but it can provide the mechanical strength. So if you don't have a mechanical the structure, then any osmotic the pressure is generated and the bacteria can explode. That's why they cannot survive. Now, breaking penicillin is one mechanism. The other mechanism we found later, they pump out those drugs. It's not breaking, it's just a pump out. Another mechanism they found, the structure of penicillin binding protein or the trans-peptidase, they change their structure. They keep their own original enzyme function, but they change those binding site of penicillin and they avoid the binding of penicillin. These three are main resistance mechanism of antibiotics. Okay, this is the key part of this talk. So this penicillin talk, we just heard the mechanism, the target is a cell wall, and beta-lactam structure is a key structure which binds to that enzyme. And this bacteria resistance against the penicillin is three mechanisms. One is destroying the beta-lactam structure. Second is a pump out the drugs. And this mechanism can be applied not only to the penicillin, it can be applied to any other antibiotics, right? So that means this can be a more broad mechanism for resistance. And third mechanism here, this is the changing of the penicillin binding protein. This is specific only to the penicillin. These three are main mechanism of antibiotic resistance. Okay, this part we didn't cover in the last class, but this is something I wanted to deliver to you as a main the lessons for this antibiotic mechanisms. Okay, now do you have any other question for this penicillin part? For now, you don't need to distinguish penicillin and amoxicillin. They are similar class of molecules. They share this kind of common structure, beta-lactam structure, and then maybe the side chain is slightly different. It makes some difference of penetration or access to the target. Other than that, these are same class of molecules, beta-lactam. Okay, any other question? If not, we move to next talk about the vancomycin. So this is also targeting cell wall with different mechanisms. Jungwon, would you like to show the vancomycin story?
Wait a minute.
Okay, I will stop my sharing. Okay, now you can share your file. Okay. Yep.
Hello, my name is Choi Young-won, and today I will talk about the antibiotics named vancomycin. Vancomycin was first discovered by the Eli Lilly company, which is a pharmaceutical company in the Borneo jungle. They exactly found the bacteria named Streptomyces orientalis in the Borneo jungle, and they got vancomycin from it. That was the discovery of vancomycin. And let me give you the brief explanation of vancomycin. Vancomycin structure is firstly looks like this, a little bit complex, and it is effective for gram-positive bacterial infections. Then, what is gram test? Simply speaking, gram test is the to dye bacteria. At first, it dye bacteria as purple and then bleach it, and then it again dye it as pink. After 3 processes, bacteria usually have 2 colors, purple or pink. If they have purple as a color, they are called gram-positive bacteria, and if they have pink color, they are called gram-negative bacteria. Vancomycin is a very strong antibiotic, so when you take this, you can have side effects such as bone marrow, low blood pressure, and hearing loss. So when people take this, they should be careful about it. And then I will talk about the mechanism of vancomycin, and the lecture was already explained some terms briefly, so I omit the explanation of some terms. Usually, vancomycin is effective for gram-positive bacteria, and thus vancomycin kills the gram-positive bacteria by inhibiting cell wall synthesis. Gram-positive bacteria cell wall is usually composed of peptidoglycan, and for the remodeling or synthesis of cell wall, they prepare the building block of peptidoglycan. Those building blocks are the NAM and NAG with peptide bond, and you may be confused about the NAM, NAG, but you can just think of it as carbohydrates or another. And they look like this. This is first one is N-acetylmuramic acid, NAM, and the second one is NAG, N-acetyl glucosamine. And the first picture for cross linkage is professor already explained, so I omitted. So the building block of the peptidoglycan get inserted, and after the cross linkage between building block is completed, the cell wall is remodeled or synthesized. Vancomycin inhibits this process by firstly, before the building blocks get inserted into the cell wall, they bind to peptide, terminal amino acid of the peptide chain, and if the vancomycin binded building blocks get inserted into the cell wall, there cannot be glycosidic bonds or the peptide cross linkage. So trans-peptidase cannot bind to peptide because of vancomycin, and glycosidic bonds are also can't be made. And glycosidic bond is also simple. It just means the bond between carbohydrates, such means NAM bond with NAG or like that. The thing is different from the penicillin is that penicillin it binds to trans-peptidase that is the enzyme for the cross-linkage of peptide, but vancomycin binds to peptide, not the enzyme. And I will give you the animation of this process because so much information was given and this is a little complicated. And process is like this. And before the intrinsic resistance, let's talk about the this part first because this is the resistance that we studied all the time. Enterococcus is the bacteria that is killed by vancomycin, but that unfortunately vancomycin resistant enterococcus was discovered. They can be resistant because of the enzyme due to their specific special DNA compared to other entities in the same species. Those enzymes can make it possible to change the structure of terminal amino acid of the peptide chain. Actually, the original terminal amino acid of the peptide chain is diala, and you may be confused about the term, the D, what it means, but it is not important for this presentation. But if you are curious about it, you can ask it after the presentation. Also, some VRE or vancomycin resistance enterococcus can change diala to diala-Dirac or diala-Dicer. For diala-Dirac, the hydrogen bonding site is one hydrogen bonding site is gone. These blue ones is cannot hydrogen bonding with vancomycin, and for the diala-Dicer, steric hindrance goes out. Steric hindrance is something like this. When molecules and molecules are bonded, there should be atoms that are around the atoms that are bonded, and if the atoms that around the bonding atoms are bigger, there can be repulsive force. So to speak, if molecule size is bigger, there can be more repulsive force, and this is what steric hindrance is. And for diala-Dicer, this site contribute to the steric hindrance. Higher steric hindrance or lower hydrogen bonding can lower the bonding force between the vancomycin and terminal amino acid of the peptide chain. By this way, this enterococcus can be resistant to it. And for this case is the intrinsic resistance means that it's not the stuff that we studied this week. This is the case that the majority or the majority of the entity in the species is resistant for the bacteria. for the vancomycin. So the antibiotics vancomycin is not used at first for these bacteria. There are several gram-positive bacteria, and those are gram-negative bacteria. Fortunately, those gram-positive bacteria is known to not be harmful for humans. Then the main thing is that why vancomycin is not effective for gram-negative bacteria. The answer is the different structure of the cell wall between gram-negative and gram-positive. As you know, vancomycin is effective for the peptidoglycan. For the gram-positive bacteria case, the cell wall is composed of a thick layer of peptidoglycan, but for the gram-negative case, it has a very thin layer of peptidoglycan, and the outer part of the cell wall, actually it's not cell wall, just cell membrane, outer membrane and lipopolysaccharide. Outer membrane is composed of phospholipid.
Sorry, Youngwon, we are out of time for now, so let's
Yeah,
let's stop here. Would you
okay.
withdraw your file? I wanted
Yeah.
to show my file. Okay, thank you very much, Youngwon, for the good presentation. Now here, to summarize for the vancomycin. So vancomycin structure is comparing to penicillin. It's a huge. Penicillin was only size of this molecule. You can see the screen, right? So only this size. Now the vancomycin is huge. That's why the binding, originally penicillin binding is to the enzyme which make this linkage here. Vancomycin itself is binding around here. Then it blocks the synthesis, the linkage here or here. So vancomycin's approach is a little different. It's not enzyme inhibitor. It's just binding to this side and then blocking here. Youngwon tried to show some of those gram-negative, positive. Positive bacteria has an inner cell membrane and cell wall outside. Gram-negative is interestingly cell membrane, thin glycopeptide layer, and another membrane outside. Then huge molecules cannot penetrate this. That's why vancomycin can work only for the gram-positive. One correction for Youngwon's, both gram-positive and gram-negative, they can be toxic to us. So we need to kill them both. And some vancomycin can work against for this, but it does not work for the gram-negative. Now the mechanism of resistance, they change the structure of the sequence here. And then vancomycin cannot bind to this structure anymore. So that's kind of another mechanism for vancomycin. For penicillin case, penicillin binding protein change this structure, and that's why penicillin cannot bind. It's similar to this. Vancomycin binding site is changed, then they can achieve that resistance for that bacteria. Okay. Now this is the difference of the two the materials. So let's wrap up for today. Texture, sorry for today. You don't have enough time to present. Maybe I will give you the chance sometime later. So next lecture will be the Tuesday, next Tuesday, and deadline is the same way. For the quiz for the website is by Tuesday, and assignment is for the Wednesday. Now this week I realized if I just give you next week the assignment is next week the topic is antiviral virus drugs. Okay, in video materials, some of those are the difference between antibacterial and antiviral drug the mechanism, and the assignment is similar to this week, but not for the antibiotic. It's for antivirus drug. Okay. Now to choose those the antivirus drug, there are many antivirus drug. Not to make an overlap. So in this assignment, you will see the discussion part. You choose your own the target of an antivirus drug. Then first come, first served approach. If you give them name, other people cannot take that drug. Okay, so you choose, make that list of the different antivirus drug, and then we will have 7 different presentations. And then depending on those mechanisms, and I will choose those similar mechanisms, and we reduce it into one, and then select as much as of different the mechanism style. Then we are going to discuss together for next week. Understood? Okay. So I opened the next week lectures already on the website. Then you look up And then if you do not understand how it works, then let me know. Okay?
Okay.
Okay, if you don't, do you have any urgent question? Urgent question in Korean is also acceptable. If it's okay, thank you very much for the presenters. Sorry, Tech June, for today. You are ready, but yeah, let's have another chance. Okay, guys, then see you next week.
Thank you.
Bye.
Thank you.
Thank you.